Hello, my name is Alison O'Mahony and I'm the VP of Translational Biology at Eurofins Discovery. Eurofins Discovery is one part of the Eurofins Scientific Organization, a leading international group of laboratories providing a unique and comprehensive range of analytical testing services to the pharmaceutical, biomedical, environmental, food and consumer products industries, as well as to governments and academic institutions. We have a large global presence and a focus on delivering the highest quality data with expert analysis to our clients. Eurofins Discovery is comprised of 7 industry leaders, many of which are recognizable in their own right as best-in-class service and product providers, with deep-rooted expertise and experience. All of these offerings are now integrated into a comprehensive portfolio to offer our clients a complete single-source solution to support their drug discovery needs. With extensive capabilities and the technical expertise of our global team, we offer the broadest scope of services and products in the industry. Service offerings include medicinal and synthetic chemistry, in vitro pharmacology, cell-based phenotypic assays, safety pharmacology, ADME tox, in vivo safety and efficacy, and an extensive menu of products. We offer multiple interactive formats for our clients, including individual projects or more ongoing relationship-based studies. Clients can also engage with us in a more integrated and cross-portfolio manner. This is perhaps best illustrated by our hand-in-hand partner model offered by DiscoveryOne Integrated Drug Discovery Service. DiscoveryOne is designed to leverage our teams and technologies to provide premier support to our clients. Using integrated project management, deep scientific expertise, global state-of-the-art facilities, and a broad scope of in-house capabilities, we can drive the accelerated delivery of a preclinical candidate for our clients. In today's presentation, I will focus on the Eurofins Discovery phenotypic services. Perhaps in its truest sense, the goal of drug discovery is not regulatory approval, but clinical adoption. Eurofins Discovery is laser-focused on helping our clients to discover a successful drug candidate that will ultimately benefit patients. To achieve this goal, a candidate must meet 3 criteria. It must demonstrate efficacy in modulating disease biology. It must be well tolerated by patients. And thirdly, it must exhibit superiority over therapies already in the clinic. Meta-analysis published by Dave Swinney and others have determined that phenotypic screening is a successful drug discovery strategy. In fact, the majority of first-in-class small molecule drugs approved over a recent decade were discovered using phenotypic approaches. This is perhaps not surprising given that phenotypic assays are closer to clinical outcomes. And while target-based approaches enable rapid and high-throughput discovery of compounds, they do require some prior target intelligence. In contrast, phenotypic approaches enable the unbiased discovery of compounds that are active for biological outcomes. Eurofins Discovery Phenotypic Services enables clients to utilize standardized and validated phenotypic assays and platforms that capture the complexity of human disease biology. 3 service platforms form the core of phenotypic services: the BioMAP platform, OncoPanel, and Biomarker services. Lab operations are located in St. Charles, Missouri, along with the OncoPanel and Biomarker scientific analysis teams, while the BioMAP scientific analysis team is located in Burlingame, California. I will first present our BioMAP platform. This phenotypic platform was initially developed back in 2000 and was prompted by the need for better in vitro models of human disease to support drug discovery. Taking a systems biology approach, the objective of this platform was to provide an approach to test compounds in physiologically relevant human assay systems, and then to analyze the resulting complex biological data sets to deliver actionable and translational insights. The three main pillars of the platform are illustrated on this slide and represent the human primary cell-based assays, the proprietary analytical tools, and an extensive reference database. First, I would like to discuss our in vitro human disease models called BioMAP systems. These systems are comprised of human primary cell types isolated and pooled from multiple healthy donors. The cell types are used at zero or low passage to preserve high physiological relevance by not being adapted to cell culture conditions and with intact regulatory processes and signaling networks. These human primary cell types are plated as monocultures or co-cultures and are then stimulated with disease-relevant factors such as cytokines or other receptor ligands. These BioMAP systems are used to assess the impact of compounds and controls on the levels of clinically relevant protein biomarkers. Assay systems are standardized and run strictly according to SOP to generate robust and reproducible data sets. This data is then visualized and mined using our proprietary bioinformatics and analytical tools. This enables us to gain insights on mechanistic activities relevant for efficacy and safety, to provide guidance on indication selection and dosing, to facilitate benchmarking, and to assess for toxicity signatures. The third pillar of the platform is the BioMAP reference database. This contains activity profiles for more than 4,500 non-proprietary agents. This database contains profiles for clinical drugs, failed candidates, mechanistic or tool compounds, and other agents. The agent types represented include small molecules, biologics, antibodies, and other test articles. The BioMAP reference database thus serves as a repository of mechanistic profiles that can be leveraged to analyze the profiles of proprietary agents on behalf of our clients and to provide valuable translational insights to support their programs. Here we illustrate the results of a BioMAP profiling study using the clinical JAK2 inhibitor Ruxolitinib. This drug was tested at 4 concentrations in the BioMAP Diversity Plus panel of 12 systems that models a broad scope of human tissue and disease biology. Clinically relevant protein biomarkers measured in these systems are listed along the x-axis, while the y-axis represents the relative levels of these biomarkers following a 24-hour treatment, or longer for some systems, by the test agent. The activities are normalized to the parallel tested vehicle control, and the test agent data is plotted as shown for each concentration. BioMAP profiles thus represent mechanistic signatures that can inform on expected and unexpected outcomes. BioMAP platform applications span the drug discovery and development pipeline, from early screening to lead optimization phases, and even through to late-stage repurposing, repositioning, or combination studies. One key application is the use of BioMAP profiling for deconvoluting mechanism of action. In this case study, a candidate was identified from a phenotypic screen and confirmed to be active in a mouse model of rheumatoid arthritis. However, the candidate could not be advanced without identifying an MOA. The BioMAP phenotypic platform provided an approach to gain such mechanistic insights by comparing the activity profiles of the candidate molecule to the profiles in the BioMAP reference database containing thousands of agents spanning multiple mechanisms. Here we show the activity profile of the candidate in red, overlaid with the profile of the top similarity match from the BioMAP database shown in blue. The most similar match for this top concentration of the candidate was a phosphodiesterase 4 inhibitor, and indeed the top matches for all the candidate concentrations included phosphodiesterase inhibitors, providing strong evidence that this was the likely mechanism of action. Furthermore, while this MOA does indeed have therapeutic potential, it was a key finding for the program since there are already several PDE4 inhibitors that are marketed in the clinic. BioMAP testing is offered in 2 formats, early screening and profiling. Early screening services involve streamlined deliverables to handle larger numbers of earlier stage compounds and is designed to rapidly de-risk and differentiate chemistry based on the impacts of samples and compounds on complex human disease biology. For more advanced or later stage compounds, BioMAP profiling services provide a more comprehensive approach to test and analyze compounds, and using expert interpretation provide valuable and actionable insights to the client. To summarize, BioMAP is the largest human primary cell-based platform used by industry, academia, and government leaders to help prioritize candidates based on efficacy, to predict safety and toxicity prior to in vivo testing, and to provide comparative benchmarking data to determine clinical superiority. Next, I would like to speak about our OncoPanel platform used to evaluate anti-cancer therapeutics. This innovative, flexible, and expansive platform contains more than 300 genomically characterized human tumor cell lines, licensed from reputable vendors such as ATCC, MD Anderson, and the Riken Collection from Japan. The OncoPanel platform can be used to test small molecules, biologics, or other therapeutic candidates as either individual agents or combinations. OncoPanel studies can involve testing across the full or majority of the represented cell lines, or studies can be formatted to focus on a particular subset of cell lines. The entire cell line panel is run every 3 months, while the smaller focus panels are run on an ad hoc basis. OncoPanel profiling services have been in operation since 2000, and the large repository of historical control data is used to validate assay performance and support quality control. A large cell line bank also helps ensure reliability and reproducibility across studies. There is a broad scope of cancer types and subtypes represented in the OncoPanel platform, encompassing a range of different tumor tissues with different genetic and genomic compositions. Profiling a test agent in OncoPanel can provide insights on anti-cancer efficacy, as well as provide guidance on dosing and oncology indications. Genomic analysis can be used to guide patient stratification for clinical testing by identifying biomarkers associated with cell line responses, as well as exploring genetic signatures within cancer indications. There are a number of formats available for testing agents in the OncoPanel platform, employing either high-content imaging as well as the more conventional cell viability assay. For high-content assays, we offer both a singleplex proliferation readout using DAPI nucleostain and report data as EC50 and IC50 values, as well as a GI50 determination. A multiplex approach is also available that includes the proliferation readout along with data on the induction of apoptosis and cell cycle status. In addition to the more standard 2D cell culture assay, OncoPanel can also be used to evaluate test agents' effects on the viability of cells in a 3D spheroid format. Assay windows are typically 3 or 5 days, but a more prolonged 10-day assay has been validated for a subset of the cancer cell lines in 2D format. All assays generate datasets that are amenable to univariate genomics analysis that are included in studies with 200 cell lines or more. Publicly available datasets from the COSMIC Sanger and the Broad CCLE databases have been normalized and integrated into our internal database. This database is then leveraged to correlate test agent activity with the genomic composition of the different cell lines. In this case study, OncoPanel profiling was used to identify the response of human cancer cell lines to treatment with Nutlin-3, an inhibitor of the E3 ubiquitin ligase MDM2. On the left is the visualization of the extensive genomic characterization analysis used to identify mutations associated with either a sensitivity or resistance to Nutlin treatment. On the right, we report the differentially expressed genes associated with a sensitivity or a resistance phenotype. This analysis enables selection of the most responsive tumor types at the genomic level, which in turn can help optimize clinical trial design. To summarize, our OncoPanel platform is designed to provide standardized yet flexible human tumor cell line panels to generate translational data coupled with scientific expertise to optimally support your oncology program needs. The final section today will focus on our biomarker services. Biomarker Services provides a more flexible analytical approach to evaluate a wide variety of test articles in a range of cell systems, such as human or non-human PBMCs, and diverse biological samples like serum, plasma, supernatants, and tissue extracts. Our Biomarker Services approach enables a more bespoke experimental design to best meet the objectives of the client study. Biomarkers are measured using different technologies to detect either soluble analytes such as cytokines or growth factors, as well as cell surface or intracellular molecules. A variety of tools and technologies are available, again deployed to best meet the study goals. Biomarker Services also has a number of more established assays aimed at evaluating agents for their impact on immune cells. Example capabilities are illustrated here and include immunophenotyping as well as immunogenic or inflammatory assessments. These services utilize our large and unique bank of human PBMC from more than a dozen individual donors encompassing different genders, ages, and ethnicities. Immunophenotyping, as well as assessing receptor and intracellular cytokine expression, are key applications relevant to evaluating test agents for immuno-oncology, infectious and autoimmune diseases. From an immunological safety perspective, our cytokine storm assay can be used to determine if a test agent triggers immune cells to release a slew of pro-inflammatory cytokines or chemokines. This outcome has the potential to unleash systemic immune activation and inflammation in vivo that can ultimately lead to multi-organ failure. Other assays report on a test agent's impacts on immune cell function or activities related to modulation of pathways such as STING or toll-like receptor signaling. Additionally, evaluation of biomarkers in ex vivo biological samples using the service can provide key insights from in vivo studies. Eurofins Phenotypic Services, operating out of our Phenotypic Center of Excellence with state-of-the-art facilities, supported by our team of experts and powered by robust platforms plus advanced analytics, can help our clients navigate their way to successful drug discovery. In summary, our Eurofins Discovery single-source portfolio can help our clients progress from concept to clinic. By focusing on efficacy, safety, and superiority, we can help accelerate the advancement of early hits and prioritized candidates to marketed drugs. Thank you for your attention. If you'd like to contact me, my email is shown here, AllisonO'Mahony@EurofinsUS.com. You can also reach out to your local Eurofins Discovery representative or visit our website at www.eurofinsdiscovery.com and click on Phenotypic Assays. Thank you again for your attention.